I'm going to start recording. And so to summarize, we had a couple polls. We're having the quiz tomorrow. It should take less than half an hour, but it'll have a half-hour window between 12 noon and 12 midnight tomorrow, Friday the 10th. And right, if you haven't checked your email or gone on the website, study questions are up late. I apologize. I'm going to finish out genes and behavior, which is the other reason to put off the quiz a little bit. And then I'm going to be talking about heritability and eugenics. Yes, the next quiz will be just on the heritability and eugenics part of tonight's lecture, the stuff scheduled for tonight, and that'll be in 2 weeks. So we were talking about prairie voles versus meadow voles. Prairie voles have all these interesting behavioral differences. They pair bond, they rear their young together. They also are more sort of social and affiliative with each other. And the hormone that moderates that a lot of these behaviors is vasopressin, which has demonstrated effects on aggression and affiliative behavior, parental behavior and attention to offspring, and pair bonding and affiliative behavior between individuals. There are vasopressin receptors in the brain that translate the presence of the hormone in your blood into behavior. And so there are a number of possibilities in how vasopressin and maybe the genes for vasopressin, or maybe the genes for the receptors, could shape the difference in behavior between these two species. And actually, none of those mechanisms are the most important. In other words, vasopressin, there isn't a significant difference in vasopressin concentration in the blood. There is a significant difference, we're going to see this in the next couple slides, in the distribution of specific vasopressin receptors in the brains of meadow voles and prairie voles. And if you remember from last week, cross-fostering experiments, when you take prairie vole pups and meadow vole pups and have them reared by prairie voles, they actually behave more like prairie voles, which is an indication that there might be something other than the sequence of the genes coding for vasopressin, or even for the receptors, that's different, because that's not something you can change by cross-fostering. So we're going to focus on the vasopressin receptor 1A gene. And the main take-home from this slide is that the genetic sequence is 99% identical and apparently functionally identical. So wait 1 second, I just want to be able to draw on things. Let's find my cursor. Here we go. So this part, the coding gene between Ochragaster, which is the prairie vole, and montanus, is the same. What's different is that Ochragaster, the prairie vole, has a repetitive expansion. In other words, it has more copies of the promoter in that's upstream of the genes coding for the vasopressin receptor. Okay, so it is more likely to express that gene and make vasopressin receptors. And then so what happens is that um, the regulation of transcription will influence how much of the V1A protein is produced, and that will influence how many vasopressin receptors are found in parts of the brain in prairie voles versus meadow voles. And it does. So if you take mice and transform them to give them the prairie vole V1A gene and promoter, and then you slice their brains and image them, you will find patterns of vasopressin receptors in the brain similar to prairie voles. You'll find a higher density. And so that's what we're looking at in the lower right-hand corner graph. So in each of these, you have a bar that shows wild-type mice. And this is the distribution of vasopressin receptors in different nuclei of the brain. And so wild-type mice have this vasopressin V1A receptors in, I think it's lower hippocampus, DB, and LS, these three brain nuclei, but not ND stands for none detected in these areas of the brain. In contrast, meadow voles, and remember this is the ochrogaster. Sorry, I said meadow voles. Prairie voles, Microtus ochrogaster, they have vasopressin receptors in every nucleus that's being imaged, right? So they have them everywhere. And when you transform the mice with the V1A promoter, the transgenic mice, which are the striped boxes, they now have the prairie vole distribution of vasopressin receptors. So that's kind of cool. What that shows you is that the difference is not in the coding gene, it's not in the amount of vasopressin. It's not in the form of the receptor. It's in the distribution of vasopressin receptors in the brain, which is affected by the promoter. But actually, transcription of that gene is affected both by the promoter and also by environment, which probably influences how exposed that promoter is and how effective it is, right? Because the cross-fostering experiments also yielded behavioral effects, which we can assume were affected by the vasopressin receptors. And why do we assume that? Because in the mice, not only did they find more vasopressin receptors in more brain nuclei, but they also showed a behavioral effect. There was more affiliative behavior on the in the mice when given when they were transformed genetically and vasopressin was presented. So my understanding is mice normally have less vasopressin, voles and generally have more, but what separates the prairie and meadow voles is the distribution of those vasopressin receptors. So that's an amazing story. However, the vasopressin receptor promoter, the V1A promoter that affects how the same gene is transcribed, is present not just in prairie voles, but it's present in some other species of voles that are not monogamous. So that means that this promoter gene might be necessary, but there's something else going on, but not sufficient. To explain the difference in behavior, right? Because if it was just the V1A promoter, then every species of vole that had that V1A promoter that led to more receptors in the brain would um, would show the same behavior, but they don't. So that's kind of interesting and a little bit sort of mysterious. Um, okay, so one thing is that the Betty McGuire studied prairie voles and meadow voles for quite a while, but there are many species of voles that have not had their mating behavior and their pairing behavior well studied. So it's possible that that vasopressin receptor is having an effect on partner preference or affiliativeness, but not sufficient to make a monogamous social system, or there has to be some interaction between the vasopressin promoter and other genes, or maybe it's something like population density or some other environmental factor that has to interact with um, the the promoter to actually lead to monogamy. Okay, so take homes of kind of the whole genes and behavior lecture. We had many different examples, many different ways of studying the role of genes, from the earlier ones like do an artificial selection experiment and see if you can affect the behavior, then it's caused by genes, or compare a bunch of species and see if the most closely related ones also share the same behaviors, um, which is especially powerful if you can actually factor out things like um, shared environments, similar ecological challenges. Then that would show that genes are affecting a trait. And then more earlier genetic methods like inbred lines or crosses, um, common garden experiments, and then more modern approaches like QTL loci and um, knockouts or RNAi to look at the effects of individual genes, um, and expression mapping. Anyway, the take home is that most traits are affected by many genes. And this is called epistasis, epistatic effects, where many genes interact to affect a single trait. And you especially saw that in like the pollen versus nectar foraging honeybees, where there are hundreds of genes that are being turned on or turned off, or when they sorry, that wasn't pollen and nectar foraging, that was making the switch from nursing to foraging in bees. You see how many genes are being turned off and turned on associated with that change in behavior. And at the same time, most genes affect many traits, and the term for that in population genetics is pleiotropy. And the pleiotropy is especially important evolutionarily because it often is the genetic mechanism behind trade-offs that limit the evolution of traits. Like if you're going to have one trait, a gene, several genes might be selected for in order to support the phenotype in one trait, in one character, but their pleiotropic effects will present costs like disease resistance or longevity, might be trade-offs of expressing a certain behavior. Um, and then what I hope will make more sense over the next couple lectures is that most genes don't code for just a single version of a trait, but for different traits in different environments. We're going to look at that in a second with the heart disease and APOI gene. APOE gene, sorry. So, you always start an intro bio with like single gene effects, but they are usually sort of the exception. And this is a really nice example, showing how sort of the best gene will be, the best version of a gene will be different in different environments. So there's been a good deal of work on the role of the APOE gene and coronary artery disease, which can lead to heart attacks, and the buildups of plaques and thickening in the arteries, which, if it constrains flow enough, can lead to heart attacks and early death and all sorts of horrible things, and cholesterol, which we've all heard is bad. So this is an interesting figure to unpack. So what you have on the x-axis is serum cholesterol, and that's going to be affected by diet, and although it's also going to be affected by genes, because how quickly cholesterol is sort of processed and taken out will have an effect. But we'll think of this, we'll think of this as an environmental effect by diet. And so we can think of this as the environment that these different genes are going to be operating in. Are they going to be operating in a high cholesterol or a low cholesterol environment? And what you have on the x-axis, on the y-axis, sorry, is the incidence of coronary artery disease, right? And so if you're gonna, like, if you smushed all three genotypes together, what would you say is the relationship between cholesterol and coronary artery disease? And this one isn't tricky, so. What do we got in the chat? Positive correlation, right. So if you were to just average these three curves, one of them's a line, but they're all curves, you'd say a positive correlation. And so if you'd say, like, this is the average of the three curves, more cholesterol leads to more disease. But there's also a lot going on here. So we all have an idea that genes have an effect on the risk of heart attack, coronary artery disease and heart attack. But what's interesting is there is no good, there is no best gene. So like, there are three genotypes. I think there are 4 or 5 alleles. And so this is, you have allele 3 and allele 2. These are different, each of these are different genotypes. So which genotype do you want to have? I'll let you think about that by throwing out a poll. Why not? And I have no idea what my genotype is for this trait. I haven't had any testing of anything. So I'm going to put the 3 genotypes. Okay, and I'm going to throw this guy out there. Aha. Go for it. Which one do you want? Okay. Oh my goodness, what should I vote for? Um, well, so here's the thing. So so there are 9 people that still haven't responded, and I totally just want to throw this. This is great. We have enough to talk about. I'm just going to give you another like 10 seconds. Oh, what the heck? I will vote myself. Um, okay. So, why are you changing your votes? Goofballs. Okay, we'll stop here. Okay. So, um, I think the winner was 34. Why someone who voted for 34? Why would you vote for 34?
I read the description
Yeah.
at the bottom. It says average total cholesterol level,
Mm-hmm.
and I'm not. I don't remember exactly what cholesterol is, but I just know that you don't want it to be too high. So I saw 34 as the lowest line.
Okay, so 34 doesn't mean that you have low cholesterol. What 34 shows, sorry, I'll try to answer it raising hands. Yes, Fatima.
I was going to say that 34 shows less cholesterol and also less incidence of CAC.
Okay, so I'm going to say one thing that since we had 2 folks that said less cholesterol, remember that like 34, the x-axis is like the environment. It's going to be affected by your diet. It's not going to be affected by your genotype. I mean, it might be, and there might well be other genes that affect cholesterol level, but these lines are not showing an effect on cholesterol level. What they're doing is they're saying, given that you have a certain cholesterol level, like let's say 200, what is your odds of coronary artery disease going to be? So what the curve in 34 is showing is if you're eating a um, a high cholesterol diet, then you will have a lower incidence of heart disease than these other genotypes, right? So given a high cholesterol diet, 34 is the best. And maybe if you know you want to eat a high cholesterol diet, then, 34 is the genotype to hope for. However, let's say you are someone with excellent self-control, and for environmental reasons, you want to be vegetarian, and overeating isn't a problem for you, or you know that you also metabolize cholesterol quickly. Now if you have a low serum cholesterol, then 34 gives you the highest chance of heart rate, and you should really want to be 33. Right? So it's really interesting if you know that you're going to have high serum cholesterol, like because you know you're going to be eating a high fat, a lot of meat diet, then yeah, 34 is the one to have. You could say, like, in the United States, you want to be 34, but maybe in Japan or a country that has a lower cholesterol diet culturally, the best genotype would be 33, right? There is no single best genotype because the incidence of disease varies by genotype interacting with environment. So whenever you have what you can see here is whenever you have what these lines are called, there's a special word for them. They are called, I'm going to type this because I'm running out of space on this page, reaction norms, or norms of reaction. And what they are showing is not the genotype in a single environment, but the genotype across a range of the genotypes. Each line is its own genotype. And on the x-axis, we have a range of environments. And on the y-axis, we have the phenotype, the trait, in this case, disease. And when these lines cross, that means the highest value of the trait in one environment will be shown by one genotype, but it will be lower in another environment, right? It shows that these genotypes have a different response to the environment. It's kind of cool. So there is, I mean, I'd say, like, in the US in general, 34 probably is the one you want to have because we're a fairly high cholesterol country. But if you, first of all, like, know that you want to limit your cholesterol through diet, and maybe diet and exercise, then um, having a different genotype might be better. And much more practically, what it means is if you know your genotype, maybe you know how seriously you have to take diet and exercise, right, to create the environment where that genotype will perform the best. Okay, this table is kind of summarizing both the effects of diet on different forms of cholesterol, low density, high density lipoproteins. Also the different genotypes and their frequencies in the population, and then also which drugs are most effective in lowering cholesterol and, more importantly, lowering um, uh, coronary artery disease, which is really interesting. So these genotypes differ also in how responsive they are to different kinds of statins in controlling cholesterol, which is really interesting. And these differences in genes also explain why it's sometimes hard to show that a given drug actually prevents disease. And that's not because the drug is not effective, but because the drug interacts with the genotypes. So it's effective in some and might actually be harmful or just not effective with other genotypes. Okay, so genes influence behavior all sorts of different ways, and they have effects during development that they are affected by the environment during development in a bunch of different ways. In other words, the environment may include the womb. We are going to be learning about that when we talk about sex ratio effects on dispersal behavior. It can be social licking and grooming behavior afterwards, and obviously, we're going to be spending a lot of time on human social environments. But it also means that you can't separate the role of genes from the environment in any traits, but especially in behavior. And one last thing I want to, I don't know how statins lower cholesterol, which is bad because I have known it because my mom takes them. But it's like it went in and went out and it's gone now. One other interesting thing is we think of like genes being traits being plastic. Plasticity means plasticity ability to develop different phenotypes in different environments, right? That's plasticity. Resilience is the opposite. You show the same phenotype over a range of environments. Thank you. But actually, like, sometimes the effects of the genes have to be flexible in order to maintain the same phenotype in different environments, right? So resilience is the ability to show the same phenotype over a range of environments. And I'm just going to show some like goofy slides. This is actually a very old graph showing after this is like after a guy named, I forget what CK stands for. I think it's Conrad Waddington. And Waddington was really interested in this process, this phenomenon called canalization. And canalization are all the forces that stabilize development. You can have an embryo developing in very different environments and it still manages to have, the same bowel plan, number of toes, feet, limbs, number of traits. And so, he's sort of describing this, and that what he's describing is that over like long periods of evolutionary time, sort of different, only a narrow range of phenotypic outcomes are possible. And that's what these valleys are sort of showing us here. And then, but the environments can change and the phenotypes will change slowly, even though they are highly canalized, sort of stabilized. And what Jablonka and Lamb were arguing in, I forget if it's the chapter we read or the second chapter, is that there are a whole bunch of regulatory feedbacks that are sort of happening underneath that sort of landscape that keep development along a range of values and respond flexibly. So even though the genes should lead to very different phenotypes in different environments, the epigenetic landscape, the effects of multiple genes on traits and their regulators are going to be sort of stabilizing traits over a longer period. I wanted to give one sort of example of developmental plasticity leading to the resilience of a trait. And so I'm going to write that down because that may or may not make so much sense. This is an example of how developmental plasticity leads to stable behavioral trait. Okay, and the trait here is orientation in pigeons. But there's no reason not to think that's not general to lots of birds. So, not sure who is trying to call me, so sorry. There's just, if there's any chance that it's my daughter, I have to see what is going on here. And it is, I have no idea. It was just a ring. Sometimes a ring is just a ring. Okay. So pigeons they're great birds for studying orientation and navigation, partly because as opposed to migratory birds who have an urge to fly 600 miles, 2,000 miles, and then they're gone, pigeons have a homing instinct, and so much so that people have spent, centuries breeding them for their homing behavior and having races to see who can return the soonest, sometimes being released from unfamiliar locations that they've never been to before. And so it's a really good system for studying orientation because they keep trying to come home. This was the first project that I had a research job on. So I was like training the birds, taking them out to, oh, this is going to be pleasant, taking them out to new locations to train them and then testing them. And one of the things we would do is we would sometimes glue a magnet to the back of the neck. This is from 1971. I was working on them in 1986. So this was an old experiment even when I was an undergrad. And what you do is you sit on you have a wooden box built onto the back of a pickup truck that's sturdy enough so you can stick an office chair that swivels, and you release the birds one at a time, and you have your binoculars, and you look at the birds until they disappear on the horizon, and then you take your compass and you figure out what direction they came and you write down what direction they disappeared at. So you record their disappearing location. And then you look at a map and you see how far that direction is from home. So in these figures, each dot is an individual pigeon. And so what you can see is if you have, on a sunny day, you have a brass, this is the control. So the brass is the control. They are all oriented pretty darn close to the true homeward direction. There seems to be a bit of a bias towards the west, but pretty darn good. So here's the interesting thing. If you stick a magnet onto the birds, that's what this experiment was looking for. Are the pigeons using variations in magnetic field? If you glue a magnet onto the pigeon, they're still well oriented, no problem. However, when you do the same experiment on an overcast day, so heavy cloud cover, we had a lot of these this summer, can't see the sun, can't even see enough. The arrow represents the average of all these dots. Okay, so actually it's kind of fun when I teach animal behavior, we do a little experiment on orientation. We do it on humans, which is fun, and you learn basic statistical circular statistics. It's a very simple thing. It's just it's different than other kinds of numbers because it's 0 to 360 degrees or 0 to whatever 2 pi radians, and understand, it's the same as like averaging time. The average of 11 p.m. and 1 a.m. is not like 6.5 or 5.5. It's midnight. It's 12. And so you need to be able to use a scale that will do that averaging correctly. Anyway, those arrows are the average. And here's the thing. The length of the arrow from, is proportional to, if it goes all the way to the outside of the circle, it's proportional to how how much they're all pointing in the same direction. So to finish this example, on an overcast day, the pigeons with a brass bar on their back are still well oriented. But these guys, you see that the arrow is short. That's because there's points all around. They're actually not, they're randomly oriented. They're not directly oriented in any direction. So magnet plus overcast is confusion. So what does that mean? What's the take home of this figure? What does this figure say about the cues that pigeons can use? Is there a hand? The sun. Um, the direction of the sun. So actually, it turns out it's not the direction of the sun, although they can respond to that. They don't have to see the sun. If they see enough of it, turns out that the sun creates patterns of polarization all through the sky, and pigeons are sensitive to polarization. But they used northwest, east, south. So they are well oriented. They can use both magnetic cues and light celestial cues. And it turns out it's not just where the sun is, it's the polarization. But if you block up, and what the reason why you conclude that is if it's a sunny day, they don't care whether their magnetic cues are being interfered with a magnet or not. However, because they can use the position of the sun and the polarization of light in the sky. However, on an overcast day, now the presence of a magnet confuses them. So the technical term is they've got a hierarchy of cues. First, use solar cues. Position of the sun, polarization. But if not, whoops, and use magnetic fields. So that might be what Suleiman was referring to with northwest, east, south. At compass directions, act like a compass, be responsive to magnetic cues. Here's the developmental flexibility, resilience side of this story. So the summer that I was working on this project, we all had to take turns camping out. The main project was in Ithaca, but the researcher had started in the Boston area, and it was just known that, oddly enough, birds that were reared in this place called Codman Farm were not confused when you glued magnets to their back under cloudy or sunny skies. And you can see that here. Iron Mine Hill it's called Iron Mine Hill because there was an iron mine. The presence of iron deposits in the ground distorts the local magnetic fields. This area is a magnetic anomaly. And what it does to normal pigeons, these are the pigeons from Fox Ridge Farm, when they're released from Regis College, which is not an anomaly, they are well oriented. Both these guys are well oriented and Codman Farm birds are well oriented. However, when they're released from a different location, Iron Mine Hill, the Fox Ridge Farm birds that are confused when they're released at a magnetic anomaly, and the Codman Farms birds are still well oriented. So these guys are confused. These guys are still pretty well oriented. So the birds that were raised, and then it was discovered that Codman Farm also is located on an iron deposit, on a magnetic anomaly. So the birds that grew up in a magnetic anomaly basically learned that magnetic field cues are unreliable or not really informative about where home is. And they learned during development to depend on other cues, so that then if you release them at another magnetic anomaly, or if you glue a magnet to their back, they're going to be using other cues, even on cloudy days, which is why we were studying smell the summer that I was working on them. But this is a way where you have these developmental pathways, these interactions between genes and the environment, and they actually are flexible enough to be able to maintain the same behavior orienting to home under a range of environmental conditions. Okay. So yeah, it's true. So the pigeons that grew up on a magnetic anomaly, Codman Farm, were well oriented when released at a different magnetic anomaly. They'd learned not to depend on magnetic cues. Okay, I am going to stop the recording for a second.